Hello everyone. Today I'm talking about the paper Engineering Human ACE2 to Optimize Binding to the Spike Protein of SARS Coronavirus 2. The two proteins I mentioned here are involved in SARS-CoV-2 infection. This paper was just published last month in Science, and the researchers are putting US military funding to good use. So the problem, as everyone knows, is the worldwide COVID-19 pandemic, where SARS-CoV-2 causes severe respiratory failure, especially in at-risk groups like the elderly whose immune system cannot clear the virus. It is also extremely infectious, causing forced lockdown measures across the world. And this is an issue that has never happened before. But the actual problem, other than a lack of vaccine, is the lack of treatment. If there is treatment, the disease will not be as fatal, and you no longer need such strict lockdown measures. So to create a treatment, we must first understand how the infection occurs. The most upstream process, the first thing that happens, is the virus binding to the angiotensin converting enzyme 2 receptor, or ACE2 receptor. This the virus spike protein is the one that binds, and the binding site on the ACE2 is known. It is 117 amino acids long. This ACE2 is present in many different cell types, in particular epithelial cells that line the lungs and intestines. Hence, these organs are affected most. The virus binding to cells also inhibits ACE2 from its role in controlling blood pressure and inflammation, which I will talk about later. So the first thing everyone thinks of for treatment is to make use of antibodies. But the problem about this is it is easy for the virus to mutate during replication, allowing it to escape antibody binding. But it's still possible for it to bind to the ACE2 receptor and cause disease. So instead, what we can do is to provide the virus with a decoy or an imposter to bind to. A soluble ACE2 can be used. Since this is very similar to the actual receptor, the virus cannot mutate to escape binding. Else, it would also fail to bind to the human cells. However, treatment with wild-type ACE2 does not work very well to stop the virus. It has lower affinity than the antibodies. So the researchers want to engineer a mutant ACE2 that can bind better. So this is the workflow the researchers adopted to engineer ACE2. The first step is deep mutagenesis. Sounds deep, but I will explain. So they made a plasmid library for all possible substitution in the binding site of ACE2. So each plasmid containing one mutation. It is one of the 117 sites, one of the 20 amino acids. Hence, in total, there is 2,340 mutants, including the stop codon in this library. Next, they express the plasmids in the human cells, and flow cytometry is used to sort out the cells that bind high amounts of spike protein. Sequencing is done, and then the frequency of the mutants is compared to the frequency of plasmids. And using this, they can identify high affinity mutants. And then following that, testing can be done on the mutants to test its affinity and how it halts SARS-CoV-2 infection. So here are the results. Firstly, flow cytometry. After expression of the plasmid library is shown, each dot here represents one cell, and the amount of ACE2 and spike protein on each cell is measured as shown on the axis. The left graph is cells transfected with the wild type ACE, The right is with the library, transfected with the library of 2,340 plasmids, so containing one mutation each. And you can see on the graph on the left, there's a linear correlation where more ACE2, the natural variation in the amount of ACE2 expressed, more ACE2 means more spike protein binding, and so it makes sense. Then next they went to sort out cells with high spike protein fragment binding, as shown in this box here. And RNA sequencing is done on these collected cells. So after sequencing, this is the result of deep mutagenesis. On the right here is the full 2,340 mutants. And the frequency of the mutants after sorting was compared to the plasmid library, where blue is increase in frequency and orange is decrease in frequency. Black is simply the wild type. So to understand this, for example, if 0.01% of the plasmids contain this S19P mutant, but after cell sorting, 0.1% of mRNA, 10 times more of the mRNA and hence cells has this S19P mutation, it is higher in frequency and hence it is enriched, and it's shown here in blue. And yes, I hope this makes sense. So this is fundamentally how deep mutagenesis works. Following that, the researchers mapped the deep mutagenesis data to a 3D structure of ACE2. The spike protein is shown here on the left in green, and orange here would represent conserved residues for binding. So the spike protein will fail to bind if it's mutated, and hence the frequencies are reduced when mutated, hence it's orange. While blue is sites that can be mutated to increase binding affinity. And then white is no effect on binding because it is tolerant to mutation. And in fact, this white valley here is the catalytic site of ACE2, and it is not involved in the binding of spike protein. Here I have grabbed another image from the protein data bank, and hope it's a bit clearer. This is a bottom view that you can see that there's a huge cleft, a valley here for the function of ACE2 receptor. Then next, the researchers want to look out for polar amino acids because that gives a stronger interaction between proteins. And this diagram here on the left is colored based on whether there's a preference for polar or hydrophobic amino acid mutants. And basically the researchers are looking out for hotspots of blue that would improve the binding affinity. And then they identified two important helix at the binding site, which looks like this. And based on the overall colors of the diagram, they identified that there's mutations in this blue box that is enriched for polar amino acids to bind to the spike protein, while mutations here in the black box, it has more yellow, which are enriched for hydrophobic amino acids to pack these two helixes better, which improves binding. Then they also realized that there's this dark blue spot, and it's actually two amino acids that allows for glycosylation of the ACE2 protein. And they find that with mutations, this glycan group cannot bind, glycosylation cannot occur, And without this glycan group, spike protein binding is much more improved. And matching the data, there's an exception here for T92S mutation, which maintains this N-glycan. The N-glycan would still form, and then hence it's not enriched. So these mutations could also explain why the disease is more severe for some individuals with COVID-19. They also went on to identify some enrichment hotspots here and explained some of the mutations. So for example, there's this D30 to E mutation, glutamic acid E, which extends the acidic side chain to interact with K417, and this improves binding affinity. And they also mentioned that another paper doing targeted mutagenesis also identified this D30E mutant, providing extra evidence that this indeed improve binding affinity. And they also go on to discuss other mutations here that I would not talk about. And then lastly, they also talk about how they provided more details on mutations that would affect the overall structure of the binding site. And so there are these 5 sites that they mentioned where proline is favored. And as we know, proline has a tight angle that is good for forming protein helixes. And they conclude that these mutations listed stabilize the 2 helixes I talked about. And they also mention many other mutations that affect ACE2 structure and hence binding to the spike protein. So however, there is a caveat to deep mutagenesis, where some mutants might affect ACE2 expression. So when they test these mutants individually, they do not actually show improved binding. This is because in cases of low ACE2 expression, with the same amount of spike protein added, the ACE2 is actually saturated with spike protein. So the frequency appears higher, giving a false positive. So validation is needed. Then with some mutants identified, they now want to test combinations of mutations. On cells with the spike protein already bound, they tested different concentrations of soluble ACE2. The T92Q mutation here is one of the mutants without the glycan group, and it shows a significant increase in binding as expected. Then for the rest here, the combinations, they picked mutations that are slightly further apart to avoid side effects, and they showed that these combinations greatly increased binding affinity. But of course, there are many other unexplored combinations of mutations that could have greater effects. Finally, they settled on this variant 2 containing 3 mutations. It binds well, it expresses well, but it contains the N-glycan group. Because as exciting as it is to know that removing the glycan improves affinity, at the end of the day, the protein has to be injected into patients, and there's a need to minimize immune rejection. And the researchers are concerned that without the glycan group, the ACE2 becomes too different. So the last stage now, after identifying a good an engineered variant, is to do testing and characterization of this variant. So they first tested against COVID-19 patient antibodies using ELISA, and the 3 blue lines here show the engineered ACE2 outcompeting the antibodies, while wild type in gray cannot. They also note that they hit this 80% limit which shows that most of the antibodies actually bind at the same place as ACE2 on the spike protein. Only 20% do not. It's an interesting thing to take note of. And they did further experiments. So they did further experiments of this engineered variant. So comparing the solid blue line to the solid gray line, the engineered ACE2 binds much better than the wild type. However, they also tested this ACE2 fused with IgG1 antibody, shown in dotted lines. In this case, the engineered ACE2 doesn't show much more improvement than the wild type ACE2, because for this fusion, there are two ACE2 molecules fused to the IgG1, and hence they conclude that avidity plays a large role, and not just binding affinity. So to further test this, they competed the engineered and wild type ACE2 against the IgG fusion with a wild type ACE2. And although the IgG fusion, shown here in gray dotted, is better than the wild type ACE2, the gray solid line, even it requires like a large amount of wild type ACE2 to be added for it to outcompete this IgG1 fusion. The engineered monomer, shown here in solid blue, is still able to outcompete this wild type IgG fusion. So avidity is important, but affinity still matters. Both are needed. So to make use of avidity, they made a dimer of the engineered ACE2. Normally, ACE2 exists on the cell surface as a dimer, so they just needed to include this dimerization domain into the protein. Previously, in the experiments, they were only using this top bit of the receptor. Then testing this dimer on patient antibodies again using ELISA. Comparing the wild type dimer in gray and the engineered dimer in purple, the engineered ACE2 competes better with patient antibodies for the same 3 patient antibodies used in the previous experiment. Next, to characterize the binding affinity of the dimer, they use biolayer interferometry, and they got the dissociation constant of the wild type dimer to be 22 nanomolar. While for the engineered dimer, it has a KD of 600 picomolar, which is much stronger than wild type dimer and comparable to the KD of patient antibodies. Now, the last step is to test in the biosafety level 3 room to see if it can really block the infection of cells in cell culture. With a lower concentration, here we see that the engineered ACE2 monomer prevents infection compared to the wild type, as seen by this solid and gray line. However, the dimeric wild type ACE2, shown in dotted gray, performs much better than wild type, which actually, as expected, it's because of avidity. So meanwhile, the dimeric engineered ACE2 performs the best, and it starts having an effect even below 1 nanomolar, and this is very promising for treatment. And also, it is interesting that this can neutralize SARS-CoV-1 even without engineering for SARS-CoV-1. And that's the SARS outbreak that we experienced in kindergarten back in 2003. So this is important, as it provides potential for future coronavirus that has not crossed over to humans yet, touch wood. Lastly, this soluble ACE2 treatment can have dual function. Normally, ACE2 regulates blood pressure, inflammation, and wound healing by cleaving this angiotensin II hormone. The virus blocks this activity and that causes inflammation, high blood pressure, and injury to organs, which makes COVID-19 more severe. So by applying soluble ACE2, it can help to rescue by cleaving angiotensin II. The researchers showed that the soluble ACE2 is functional, although at 25% lower activity, and it's not hindered by the virus since it binds to a separate site than the catalytic site as mentioned previously. And there's also no cell membrane now blocking the soluble ACE2. So just some thoughts on the research. I was thinking whether will this soluble ACE2 treatment prevent immune response since most of the antibodies bind to the same site as the ACE2, and whether or not now with this treatment you cannot have your antibodies binding to the spike protein to have a appropriate immune response. But that might be okay since this treatment should be used in severe cases where the immune system is not responding well anyways. And then another question I had was why not to use the ACE2 IgG1 fusion instead of making like a dimer. And in the first place, I don't really understand why the IgG1 fusion was tested. And then as mentioned previously, there's a 25% lower catalytic activity of the soluble ACE2, and this might be a good thing since we don't want too much angiotensin II cleavage. But yeah, only future trials will give us data about whether this is helpful or not. Then for future work, it's good to think about whether this dimer is able to enter well into organs and whether it is stable in blood. And lastly, I have an idea that whether it's effective or not if we use a mixed population of different mutant ACE2 that will make it harder for viral escape to occur. So in summary, this research provided insights into the protein interactions necessary for coronavirus invasion into human cells, and it has shown how high affinity can be engineered into the natural ACE2 for binding to the virus. And this serves as a promising treatment for severe COVID-19 cases. Thank you so much. Thank you everyone for watching. Please leave your questions and in the comments section, and see y'all.